The Life Sciences DNA podcast is sponsored by Agilicium Labs, a collaborative space where Agilicium works with its clients to co-develop and incubate POCs, products, and solutions. To learn how Agilicium Labs can use the power of its generative AI for life sciences analytics, visit them at labs.agilicium.com. Amar, we've got Manfred Stapff on the show today. Who is Manfred?
Manfred is a physician and pharmaceutical executive specializing in internal medicine, clinical pharmacology, and data-driven healthcare innovation. He has held leadership positions at major pharmaceutical companies such as Merck, Forest Labs, and LG Chem, where he managed global clinical development, regulatory affairs, and medical operations. He served as chief medical officer at TriNetX, helping build the largest real-world data research network in healthcare.
And what is Candid Advisory?
Candid Advisory is a consultancy firm founded by Dr. Stapff, specializing in AI-driven health analytics and regulatory strategy. The firm offers guidance in health analytics, drug development, regulatory affairs, and the strategic use of real-world data for clinical research and decision-making.
And what are you hoping to hear from him today?
Manfred has written and talked about the need to combat misinformation and advance evidence-based medicine. This is a very real problem in the realm of healthcare. So I'd like to hear about how we can do a better job in getting the real-world evidence and how AI can really help drug developers, regulators, policymakers, providers, and patients to make better decisions based on real-world data and real-world evidence.
Before we begin, I want to remind our audience that they can stay up on the latest episodes of the Life Sciences DNA by hitting the subscribe button. If you enjoy the content, be sure to hit the like button and let us know your thoughts in the comments section. And don't forget to listen to us on the go by downloading our audio-only version of the show from your preferred podcast platform. With that, let's welcome Manfred to the show.
Manfred, thanks for joining us today. We're going to talk today about life sciences and how real-world evidence and AI can help various stakeholders in our healthcare system to make better decisions. We're in the age of social media where information and misinformation spread quickly. There are times today when it seems like facts don't matter. We can think about the COVID pandemic and the controversy around vaccines. How do you view the information landscape today? What has changed, and has misinformation become a public health threat?
Yeah, first, thank you very much for having me. I'm really glad to be here and also to exchange opinion and to provide my messages to the audience or to the world, I have to say, because this is really interesting time. The information landscape, you're absolutely right, is changing dramatically. What I found out is that it became dangerous if we fail to correctly deal with it, if we cannot handle it. The reason being the change in speed, in technology, in how information is distributed, obviously, as we know all, has accelerated dramatically in the past 5 years. But the speed of our brain to digest this information and also the speed of the regulatory system to regulate and the legislative system to kind of regulate this information flow has not accelerated that much. And out of this discrepancy between how we can deal with the information and how quickly the information spreads and how slow the information regulation is changing, this can become a threat. But it also, if we deal with it correctly, it can be a huge package of opportunity. So I'm sure we will go a little bit more into the details, but this is what I see as the biggest impact, the difference in speed of spreading information versus digesting information.
Okay. And how, so before we dig down into Candid Advisory, what role do you see AI analytics and their application to the real world evidence as helping address this threat of misinformation?
I mean, AI is a wonderful tool if you use it correctly, and if you don't abuse it, absolutely, because again, it's the speed and the volume which AI can digest compared to our brain. So, as a research tool, it's a wonderful help. But AI can only digest or analyze the information and the data which it gets, and usually it screens the internet for information. So if the information on the internet is somehow biased in a certain political direction, a certain racial direction, in a certain scientific direction, then the output of AI will also be biased. So this is something definitely which we have to keep in mind. Also, pharmaceutical companies, if they apply AI to their clinical research data, then the output will be also have the same non-representativity and the same experimental character as clinical trial data have as compared to real world data. So again, it's a wonderful tool, but we have to use it correctly.
And in the world of, and before we go to the clinical and the real world data, in the world of information, how do you regulate that? Because as you described, right, AI is using information that it's being trained on, but how do we make sure that it is being trained on the right information or right data? Like, how do we keep an eye on that, or how do we manage that?
If we can influence, obviously, we should be aware of that. And the example of the pharmaceutical industry analyzing their own clinical trial data is a typical example where we can influence, meaning we can limit or define the data volume, the information volume, which the system, the artificial intelligence gets. In a case like ChatGPT, we do not have an influence. We can certainly ask questions, and by asking the questions, also tell the system, I want to see this type of population. I want to see this type of science. I want to see this type of publications coming from that area. So by asking the correct questions, we have a certain influence on which information, for example, ChatGPT will be digesting. But overall, we have no influence on what ChatGPT sees in the World Wide Web. But again, asking the correct questions may help us to push it into a direction which is perhaps less biased, or also push it into a direction which is perhaps more biased. We are not completely powerless, but let's be honest, ChatGPT, of course, does what they can do, and is doing what they have been trained to do.
Yeah, absolutely. So in the pharmaceutical world, as you mentioned about the clinical trials, what are can you talk about what are some of the limitations of clinical trial data, or like why is that not good enough?
Clinical trials are an experimental system. We have a study protocol, we have defined visit intervals, we have defined inclusion and exclusion criteria. We have basically an optimized population, optimized for efficacy and optimized for safety. It has nothing to do with cheating. It is simply for scientific quality in order to evaluate efficacy in the best way and the best scientific correctness. We want to define the patient population in the best way possible. And for safety, also for the safety of the study patients, we want to exclude patients who have a certain risk. Elderly people, pregnant people, lactating people, we all know these exclusion criteria. The problem is then when we move forward after phase 2, where in phase 2 we want to have this scientifically super correct population, Very often we have a copy and paste situation from phase 2 to phase 3, and this idealistic patient population continues to be enrolled in phase 3 and has not so much to do with reality. So the exclusion and inclusion criteria need to become much more wide, much more liberal, much more closer to the characteristics of the population who will later take the drug once it's on the market. Our own analysis showed that in most studies, the study eligibility criteria restrict the population to approximately 10% of the population who will later have the indication to take the drug. Vice versa, this means that for 90% of the patients, because of the various combination of in- and exclusion criteria, for 90% of the population, there are not enough efficacy and safety data. So if I was sitting at the FDA, I would give a pharmaceutical company all the exclusion criteria of their phase 3 studies as a contraindication and would say you don't have data. It happens partially, or you don't have data on kids, you don't have data on this, but it's much more a matter of representativity of the population. And we know partially also from more or less political and ideological discussions, minorities are underrepresented. Study patients are more often white, are more often male, are more often young, have more often a higher socioeconomic status than the real population who has the disease and supposed to take the drug when it's on the market. So in this case, the ideologically kind of tainted discussion comes very well together with the scientific discussion. Yes, minorities are underestimated in clinical trials. And I was now only talking about the population. Now let's talk about the procedures. We have a study protocol with visit intervals every week, every 2 weeks, every month, controls, checks, double checks, special treatment. If you're study patients, perhaps you do not have to wait in the waiting room for hours like other people in the emergency room. So no patient gets treated in a real world situation so well and with so much care as study patients are treated in a clinical trial situation. The consequence is non-representative population, placebo effect, and Hawthorne effect. Hawthorne effect is the effect that if people know that they are observed, they behave differently. In many studies, there are questionnaires to be filled out. So if I know because I have to provide my informed consent, of course, I may behave in a different way than I would behave in reality. So this all makes randomized clinical trials a perfect scientific tool, but it needs to be complemented by real-world data, and both results they don't have to be identical. The results don't have to be a total match, but they should point into the same direction, because clinical trials are an experimental situation, artificial situation, and very often have not too much to do with reality.
So one of the arguments that the pharmaceutical companies make, especially about like when you have the heterogeneous population, the statistics becomes harder. You need to have larger sample size, right? So that increases the cost a lot. That increases the study time a lot, right? So what would you say to that?
A sample size alone will not solve it. Just looking at the representativity of the population, if the population is too young and too male, you can increase the sample size and have a false sense of certainty, because by increasing the sample size, you only increase the statistical significance, but not necessarily the clinical relevance.
Okay.
So it has to be complemented by real world data.
Okay. So let's talk about real world data. So the term real world data and real world evidence, right? These are related terms. Can you, like, for the audience members who are not familiar with these, can you talk a bit more about what these mean?
Yeah, the definition of real world data I like the most, the FDA definition. It's funny, they define what it is not rather than what it is, but this is easier. So real world data is everything what does not come from an experimental situation, does not come from a clinical trial, does not come from any kind of scientifically impacted or controlled situation. This can be whatever data come from a day-to-day interaction between doctors and patients in the hospital, can also come from smartwatches, smartphones. We have a lot of health-related digital health applications, measure heart rate, can read the ECG, some even can or pretend to measure glucose values, oxygen measurement. So it can come from all situations which are not influenced by a by a scientific study protocol of experimental protocol.
Okay. And what about real world evidence, which is related? But like, so when you
Yes.
talk about that,
So
yeah.
thank you for triggering that, because this first thing on the real world data. Real world data can only become real world evidence if they are collected well in high quality and analyzed with proper statistic methods, for example, without bias and with cross-checking for outliers, for example. But also if the interpretation of the statistical analysis is done in a correct way, similar to clinical trials, and then also when the interpretation and the conclusions are communicated in a proper way, adjusted to the respective audience. So real-world data alone are not the solution yet. It's what we make out of the data by proper analysis and proper communication.
So real world data by definition is not going to be as clean as the clinical data in the well-defined experiment, the clinical trials that the pharmaceutical companies do, right? So real world data is going to have a lot of, as you were talking about, population is going to be heterogeneous. The way people take drugs is going to be different. Not all the tests that are neatly done in clinical trials, that's not going to be here. So how different do you usually find analysis of real world data compared to the analysis of clinical trials? And what are some of the challenges that you find with the analysis of real world data?
So one thing is obviously bias, because it's not randomized in the real world clinical trial world. Is a bias. So things or changes which we see in the overall course of a disease may be explainable also by the reason why a patient got this drug. Some very simplest explanation. If a specific drug, for example, is preferably given to elderly or to more sensitive people because it has a reputation that is very safe, then of course we have a bias in the result. I'm mentioning that because this is what basically broke the neck in the Vioxx case, because Vioxx was promoted as so safe that a higher percentage of risk population got Vioxx and then of course experienced also myocardial infarctions or other complications. And this could have been attributed to the age of the patient, but it could also have been attributed to the drug. But the conclusion is real world data, if there is no randomization, they can be biased. Therefore, in the analysis, if one compares clinical trials, which are most cases randomized, and one compares them with real world data, one has to take this into consideration. The demography may be different, the concomitant therapy may be different, the risk profile may be different, and this has to be considered, for example, with propensity scoring during the analysis.
Well, I was wondering about, so real world data is then removing the biases that are inherent in the clinical trial populations, right?
I would phrase it in that way. By randomization, you have less bias than in the real world data. Therefore, in the real world evidence generation, meaning the proper analysis, you have to consider that you may have this bias and you have to work on the bias. For example, identifying potential influence factors others than the medication, if you compare it with some product, and do propensity scoring to balance by age, by risk factors, by smoking habit, or by whatever is important for your specific therapy or for your specific disease.
Okay. And any other things like there that like, so we talked about bias, but what are some of the other um other areas in which real world data is different from clinical trials, like collection or like even the cleanliness? Like, can you talk
Yeah.
about some of those? Yeah.
Yeah, since there is no scientific experiment, no study protocol which requires which data to collect and when to collect them, there is no clinical monitor who does the source data control, there is no data management in a pharmaceutical company who makes sure that the data set collected is complete and perhaps goes back to the investigator if the data set is incomplete, missing data, or if there are inconsistencies, or if there are just typing errors, decimal point errors. So all these quality management steps are available in clinical studies, but not in real world data. And therefore, similar steps need to be introduced in the collection of the data. When basically the data come in, usually this is a vendor, a data broker, an organization who collects this data. These companies need to have a quality management which applies reasonable data quality tests. The most simple one is look for outliers. You cannot survive if you have a potassium of 50. You can have a potassium of 5.0. So these kind of tests, of course, need to be done for completeness. Also, if you have in the data set a treatment for thyroid disease, then there should also be a diagnosis for thyroid disease. So this is a test for consistency, completeness, and reasonable. So such quality management steps should be introduced. Similar like in a factory, when you build a car, then also the parts which come before they go into the manufacturing belt, the parts which come from a deliverable, they need to be tested for quality before they are mounted into the car. And then at the end, when the car is produced, at the end, there's again a quality check for the end product. Something similar needs to happen in organizations or companies who collect real-world data, combine them, and sell them to the pharmaceutical industry or to other organizations who are interested in real-world data.
Mm-hmm. So then what you're saying is that the data preparation for real-world data is much more difficult than when you're analyzing clinical trials, right? Because you have to go through all this. There are all these things that you need to consider that in clinical trial, because it's an experiment, those are already controlled.
It can be automated. Of course, all these checks can be some automatic checks in various algorithms. like data management in the clinical trial also is doing consistency checks, plausibility checks, and many of them are automated checks. Obviously doesn't have to be a manual work. But certainly in some instances, it makes sense if a medically trained person looks at the data set and has a little bit of an idea about consistency checks. Like I mentioned with the thyroid, there are other consistency checks where one can check diagnosis versus history, whether it does make medical sense or there are perhaps some errors in there. I would not necessarily say that these quality checks are much more effort. I'm just saying this is necessary. And then also not forgetting, when you do a real world data study, you do not have to search for patients and enrollment speed, and you do not have to wait for the therapy duration. If you are doing a long-term study for clinical outcomes for 5 years, you cannot accelerate it. If you do a real world data study about 5 years outcomes, then you need data which span over 5 years. You can do it retrospectively. So overall, of course, the costs and the effort for real-world data, real-world evidence is much, much less than for randomized clinical trials.
Okay. And as we talked about, like the incompleteness or like the cleanliness of the real world data is not that much. Does that result in not being able to use all the data or all the patient's data usually, or like how do you find that when you're doing the analysis?
That's perhaps where sample size indeed then comes into play, because if there is a high variation or missing data, this can be compensated by much higher sample size. While in a clinical trial, the statistician gives you a sample size which is necessary to work on the hypothesis with the planned p level, usually 5%. And this is usually then the number which you enroll in the study. For financial reasons, you do not want to over-enroll, but also for ethical reasons, you should only expose this number of patients in a clinical trial with an unknown drug, a drug where the safety is not yet very well evaluated. You should only expose this number to the study, to the drug, which is absolutely necessary for statistical reasons. So therefore, you keep the numbers low for ethical, statistical, and financial reasons. While in the real world, in studies which you can do with real world data, you take whatever you get, because a higher sample size gives you more certainty, not only from a statistical point of view, but also to compensate for potentially missing data or potentially erroneous data or potentially higher standard deviation.
Mm-hmm. And what are some of the can you talk about what are some of the examples of real world data? Like there's claims data, there is registry data. Like what are the different types, or EMR, right? Like what are some of the different types of real world data, and if you can talk a bit about those, like their characteristics.
Yeah, if you talk about real world data, you automatically think about electronic medical records. Thanks to 21st Century Cures Act, almost all hospitals, almost all physicians and practices are using electronic medical records. The challenge obviously is that all these data sources do not necessarily ideally talk to each other. They have different definitions, different structures. So patient linking is an important task, but it can be done by standards. And then you can connect data which come from different sources, but not only different electronic medical record sources, because a patient can be in a hospital, a patient can be in a general practitioner. The same patient can also have a visit with a dermatologist or with an ophthalmologist. So these are already situations where data need to be combined. Patient linking is the miracle word here. Then it continues. Claims data is also an interesting source, because usually you have one insurance, and then all these different data from different sources come together into this one insurance. There are claims obviously before they have been solved and after they have been solved, so there are some specifics to be considered. Then when we think about medication, in the electronic medical records, you find what medication has been prescribed, but in pharma data or in claims data, you see whether the patient actually filled this prescription, whether the patient actually went to CVS or Walgreens or whatever pharmacy and filled the prescription and at least took it home. You still don't necessarily know whether the patient then actually has taken the medication. What you do in a clinical trial, you do the pill count. It's also no proof that the patient has taken it. But if the patient brings back an empty bottle, likelihood is like higher that the patient actually took the drug. This is certainly often an uncertainty, the drug compliance. What else do we have? Yeah, then we have the patient's own digital help tools, digital health tools, smartphones, smartwatches. So all these sources. When we talk about registries, then these are already scientifically planned situations. I'm kind of hesitant to define a registry as a real world data, because registries are given to the patient by a questionnaire, so the patient knows that they will be asked certain questions mostly about symptoms and feeling and so on. So this does not really go into the real world.
Okay. So, and you were about to give an example about real world data analysis and what Candid Advisory does. So can we start with that? Maybe an example that brings together a lot of the things that you talked about.
The one example which I wanted to bring up earlier, when it comes to the difference between clinical trials and real world data, is very striking, because one has to show, and this is one thing which I'm doing in my Candid Advisory life, also looking at data and evidence and the practical examples for theoretical things which we discuss, or theoretical advice I give. And in this case, the practical example for the difference between science and real world. So a Canadian study which was published recently looked at treatments for multiple myeloma, MM, and looked at 7 schedules of treatment schemes which had been tested before in randomized clinical trials. They tested these 7 treatment schemes with real-world data and found out that in 6 of these 7 treatment schemes, the clinical trial data or the clinical trial results showed an up to 1.4 longer progression-free survival than in real world. So in the clinical trials, the treatment schemes worked better than in reality in 6 out of 7 cases. And that's a very interesting example, because it shows that, especially in oncology, where we get a lot of these studies from very specific randomized clinical trials, that these data do not necessarily realize in real world. Not surprising, the first result of this analysis was that the multiple myeloma patients in the real world data set were significantly older than in the clinical trial data set, which may also be a reason for the worse performance of the scheme in the real world.
That's very interesting. And so tell us about like maybe another example, maybe let's say on the clinical development side, how you've helped some of the pharmaceutical companies in terms of clinical trial designs, or so. Is there like an example that comes to your mind?
Yeah, classical examples for real world data is obviously learning about the patient population, learning about the disease before you even start a clinical development. What's the demography? What are the usual risk factors? What are the usual therapies? What is the patient journey through a diagnosis or through a disease? Are the lines of treatments? Very often the treatments are changed during the course. So this is a very first step which should be done. The next step is then support for protocol generation, protocol writing, in the sense of feasibility. We all know, and it goes through the pharmaceutical industry as a message since 25 years, but it's never changed, that the combination of in- and exclusion criteria is often so complicated it's very hard to find these patients in reality. Therefore, feasibility, running this protocol in silico before the protocol is finalized, to finding out what will be the screen failure rate, what may be the enrollment speed, and can you find these patients anyway? And then it continues with the potential to having an external control group if you want to compare drug A and drug B, and drug A is perhaps new, drug B is on the market. Perhaps you do not have to have the arm for drug B in the randomized clinical trial, so it would cut your trial population by half, because you can take the comparator data from the real world. It's a little bit tricky. You have to make sure that the populations are comparable, because you don't have a randomization. You still have a Hawthorne effect, because the drug B patients, they are in real world. The drug A patients, they know that they are in the study. They behave perhaps differently. So it works for some very objective endpoints where we are not very concerned about the placebo effect or other more emotional effects which impact perhaps the treatment. But it's an option which can be discussed. Then are the after post-approval requirements. There should always be a post-approval pharmacovigilance observation of the patients, of the safety profile, of upcoming side effects, of newcoming diagnosis. This should become standard, and instead of an advertising observation, which pharma relatively often uses to push a little bit the prescribing instead of these market observations, it should be just be done by collection of real world data if the numbers are high enough.
Okay. And to what extent do you see the pharmaceutical and biotech companies taking advantage of the real world data, that like before, let's say, clinical trial planning, as you gave multiple examples of where this can be used, or even post-market? Like, do you see that very common, or do you think there is still a long way to go in terms of adoption?
I think thanks to the advertising effort and promotional effort of organizations who provide real world data, the classical use cases, protocol feasibility and site selection are the most often applied use cases. Site selection also because you then know, if you check all the inclusion and exclusion criteria and you can differentiate by site, then you know where you can find these patients, at least in a geographic sense, sometimes even hospital by hospital. And these are the most often used cases. I do not see too often the general learning about disease aspect, let's say the preclinical aspect, learning about the disease in real world. As I mentioned before, what's the demography, what's the treatment, what's the standard treatment, what are the patients going through? I don't see that too often. I also do not yet see too often the pharmacovigilance part where you actively collect real-world data after launch in order to learn more, unless the FDA mandates that. One could claim that the pharmaceutical industry is hesitant to actively collect adverse event data, which may perhaps then taint their label. And therefore, it would work the best if the FDA just mandates it, or if the FDA does it, because they have their Sentinel system. So why bothering to force the pharmaceutical industry if the FDA can do it themselves?
Yeah. And what about any specific examples, let's say about regulatory strategy? How can real world data be used, or how you have used that?
Yeah, I mean, there are always confidentiality agreements, so therefore I have to tell it a little bit general without naming specific products or companies. But you just imagine that a smaller company needs external advice when it comes to develop in the United States, and the company is outside the United States. They want to develop in the United States a nasal spray with a biological compound in an orphan disease. There are so many unpredictable situations where such companies need an external advice. And in this specific case, it turned out to be so complicated that perhaps there was no return on investment. A decision to say no is also a decision, because it protects the company from a later failure. So these strategic discussions are very often coming from a desire when smaller companies, startup companies, do not necessarily have this wide expertise. They perhaps do not even have a chief medical officer, or if they have a chief medical officer, they prefer a specialist who was working in this specific disease for years or even decades. And there often come questions, strategic questions, regulatory approach questions, where someone with a wider cross-functional expertise, and I worked in medical services, in clinical development, in clinical operations in Europe, in the United States. So my experience is very wide, and startup companies have very often the desire to have employees who have a very narrow but very deep experience. So I can complement that if specific questions come up.
Yes, sounds good. And so, where do you see the biggest challenges when integrating real world data analytics and AI into the life sciences research?
One is still trust, and this is related to privacy. It's also related to quality. So everybody working in this field has to do a lot to explain what is really behind real world data. How are the companies working? Where do the data come from? What kind of quality management systems do they have? What are the analytical methods being used? And also, of course, more for the physicians and for the patients also, what data protection measures are done. Not only HIPAA compliant, or also in Europe compliance to the European data
GDPR.
laws.
Yep.
Yeah, exactly. So creating transparency, education in order to create trust is a very important step. Fortunately, the FDA has already accepted real-world data with the proper real-world evidence, obviously, and has already acted in regulatory decision-making for example, when it comes to new indications for drugs which are already on the market. Tacrolimus is one example, but also in the medical device sector, different indications for certain medical devices, simply solely based on real-world data. This is certainly a step where we can expect more to come in the future. So to the topic trust, it's really important. And this is one reason why I try to distribute trust by podcasts like yours. Thank you for inviting me. Also by writing the book Real World Evidence Unveiled, navigating through the maze of modern misinformation. So this is also not only important for drug developers and pharmaceutical industry, it also applies to our daily life. So the same way like FDA is bombarded with statements that a drug is safe and has efficacy, and they want to see the evidence, so are we also bombarded with information and with statements, with headlines in media, social media, newspaper every day, and we also want to see the evidence. So the overall thinking should be the same. And since it's a young, let's call it a young discipline, it is very dynamic, can change year by year. But now it's everything still a little bit shaky, but there's more to come, and it will become more and more accepted.
Mm-hmm. And is your book out, or is that something you're planning to publish soon?
Now the book is out. It's available on Amazon as ebook, hardcover, or paperback. And again, it says real world evidence. You find it faster if you search for real world evidence unveiled, and then navigating through the maze of modern misinformation, and then of course you find it.
That sounds great. And so now we talked about a lot of these applications of real world data in different in the life cycle of the drug, right? Where do you see it having the greatest impact near term and in the long term?
So near term, it's already accepted by safety related, when there are safety related questions, obviously. Partially by pharma, but mainly also by regulatory agencies. I mention again the Sentinel initiative. Long term, I could imagine, and this is very long term, it's almost a little bit revolutionary. I could imagine a stepwise approval process, for example, which we had with the COVID vaccine. When there was an urgent need for vaccine, there was a preliminary approval without long-term studies, with a very incomplete CMC package, chemistry manufacturing control package, because it was necessary. Now I would argue for a stepwise process, not only because it's necessary, but more because it's possible. We could have a stepwise approval process where the phase 2 studies are the classical scientific studies with a strict protocol, with a lot of in and exclusion criteria, and that these scientific studies, they will lead to a preliminary approval and allow the pharmaceutical company to bring it on the market. And then the phase 3 will be replaced by basically what we have today, a phase 4 with a post-market observation. In this case, with a mandatory collection of real-world data about this drug in the real world, how it behaves. Obviously, the patients need to know that this has only a preliminary approval. There needs to be a lot of education and transparency, but this would speed up the drug development process, would make drugs sooner available for patients who need it, would reduce the costs. Just imagine what 2 confirmatory phase 2 studies are costing. and would I don't think would jeopardize patient safety if the real world safety observation is very dynamic and reacting very quickly in case signals come up.
That sounds great. And I hope we go in that direction and we march in that direction and we can use real world data to really accelerate clinical trials, but also make sure that we have the data for different types of populations. So really appreciate that. Dr. Manfred Stapff, founder of Candid Advisory. Manfred, thank you very much for your time today.
Thank you very much for having me, and thank you for helping me to distribute the message and the facts behind real world data, real world evidence. Thank you very much.
Well, we really covered a big range there in real world data. It was interesting to hear Manfred talk about these different aspects. What did you think?
I mean, I find real world data to be a pretty fascinating area. It's something that's, of course, in the last few years, we're talking more about it. We start using a lot of real world data, but still, there's a long way to go. And both from the pharma point of view, but also from the regulatory point of view. So it was really good to see a lot of the applications that you talk about and how they're coming to age more and more. But also that I don't believe we're using real world data to its fullest potential at this point.
One of the things that really struck me was when he talked about the fact that most the eligibility criteria for most clinical trial studies restrict the population that can enroll to about 10% of the actual population that will later use a drug. We're not adequately testing drugs for 90% of the populations that will use them. What did you make of that?
I think pharmaceutical companies do have a realization that the clinical trials that they design don't necessarily cover everything. And as I mentioned there, right? I mean, there are limitations for these companies who need to show the statistical significance in a population, and it's also um, there's a limit to how many patients they can recruit. And I know most of the pharma companies are pretty active in terms of diversifying their patient population to include multiple races, to have the genders included to a large extent. So I know a lot of them are trying that, but it's something that I believe started a few years ago. And we have to see like to what extent they have now been successful in diversifying the population. So there is genuine need from the pharmaceutical companies and genuine desire to really get a lot of those populations. It's a matter of how can they do that with the constraints that are there. And so that's why if there can be if the real world data can complement in some of that aspects, that would be really beneficial.
Another thing Manfred talked about is because of ethical, financial, statistical reasons, you want to keep study sizes small. Real world data is quite different, particularly now that we can manage and analyze massive data sets. What are the implications of that?
So with real world data, yes, you can get a lot of the retrospective stuff, right? But for a lot of regulatory approvals, you do need not the retrospective, but the more like you define the specific population and you do trials. So it's much more prospective than retrospective. Also, one thing about retrospective is that you can understand what is happening in a disease area in terms of the treatment landscape of the drugs that are already on the market. Whereas when you're doing the clinical trials, there's a new drug that will be coming across, right? So it's like, the real world data becomes much more helpful for a specific drug once it's on the market, because that's when the data becomes available and you can get the retrospective aspect. So yeah, there are some limitations about it, but again, to really understand the market or like some of the applications you talked about, like the site selection or really understanding what are the inclusion exclusion criteria that can benefit and that can only harm the clinical trial, because you really just cannot recruit patients, and that's a real problem. Like sometimes the those criteria can be so stringent that it's hard to recruit, right? So using real world data for some of those makes a lot of sense to me. And then, but I'm not sure if a lot of the companies do that consistently. And as we talked about, right? Because I mean, of course, doing the real world data, the analytics, that also is an expense. And so I would say the bigger pharmas with more money can tap into that much more than the smaller biotechs who, as he talked about, they have people with more, maybe more the narrow skill set in a specific in the specific indication that they're going after, rather than the larger, broader view, or the even the means to do those kind of analysis, to buy the data, to have people who can clean up the data and do the real world analytic. There's definitely, I see the gap between what is really possible and what is the ideal versus where we are.
You talked about electronic health records as one of the primary and obvious sources of real world data. And you talked about some well known challenges that have to do with the structure and the variability of electronic health records. One of the things I'm hearing, though, is the problem with these records from a research point of view is that they're really designed to satisfy the needs of insurers and may not be well designed for research purposes. Do we need to think about changing that to make them more useful?
We can. The question is how, right? Because these are the AMRs. They're usually managed by specific health systems, and they're capturing a lot of the data so that their doctors have can see the history of the patients. Of course, what they want to know is, well, what were the diagnosis, what are the what was prescribed. But as you were talking about, right, like yeah, the prescription information is there, but whether the compliance is not necessarily there, because that's something that maybe then just doctor having conversation with the patient rather than something that gets recorded. And that's something that when we're looking at for like the pharmaceutical purpose, right, we really need to know how, like, what is the compliance, how much is that really being used, because then you really understand the effect of the drug if the patient is taking them. So it's just different purposes. But on the other hand, I understand that it's if the pharmaceutical companies start getting into the EMRs, people are going to get worried about, okay, well, what are you trying to get from those, right? So it's different people who are making sure that the EMRs are there, and then the purposes are different. So yes, there's a need to gather a lot of this information, but I don't see how that is going to happen in reality.
You asked about to what extent biopharma companies are adopting real world data. Manfred mentioned site selection as an example where they're probably most aggressive about this, not as much so on the preclinical research to understand a disease, or on the other side, pharmacovigilance once they're on the market. But why do you think that is? Is it a matter of access to adequate data? Is it cost, or is it something else?
When you want to do the clinical trials, you want to do them very fast. You want to accelerate those, and maybe in that, this might be falling a bit sideways in terms of uh, it's taking the time to get the data, buy the data, to do the analysis, to really understand. So I think one of the reasons is time. Another one is, of course, resources. Another one could be the trust, right? Like they don't necessarily know what is it that can come out of the real world data. So maybe people and these companies need to spend some more time in terms of understanding what is the value they can get from real world data. So that's why some like having these kind of conversations is helpful, so the companies do understand more about these, and then can make use of those. But again, it's more about, as we talked about, right? This is a new field. This is a burgeoning field. This is like 10, 15 years ago, the use of real world data was tiny, whereas right now it's much more. And the companies have started understanding it, but it's a journey. We're on a journey, and I believe we will continue to be on that. And companies will use real-world data more and more as time goes.
Well, I'm glad we're able to have Manfred on the show to talk about this. It's an area we haven't really explored in depth before, so that was a great conversation, and thanks as always.
Good to be here, Danny.
Thanks again to our sponsor, Agilisium Labs. Life Sciences DNA is a bi-monthly podcast produced by the Levine Media Group with production support from Fullview Media. Be sure to follow us on your preferred podcast platform. Music for this podcast is provided courtesy of the Jonah Levine Collective. We'd love to hear from you. Pop us a note at danny@levinemediagroup.com. For Life Sciences DNA, I'm Daniel Levine. Thanks for joining us.